All right, good morning, everyone, or good afternoon, depending on where you are. Thanks for coming to this meeting. I think it's tremendously exciting and also important. And just to give a little bit of background about why this meeting is happening, Clara and Leah with Ilya and his team, Violetta and Tomas, and also with a lot of support from Frank Hillary and many of you, have really been developing a consensus workflow or steps towards a consensus workflow for resting state fMRI processing in ENIGMA. And just to give a little bit of background, there are now over 15 clinical working groups that are using HALFpipe, which is a great credit to Leah and Ilya and their teams, also to Henrik Walter and Susanne Erk for supporting it. And also to Frank Hillary and Emily Dennis, who are supporting the work through their NIH grants. So what had happened is that a lot of the ENIGMA clinical groups were running things slightly differently. And I mean, obviously for different disorders, it could make sense to look at different systems. But I'm talking about, settings that were even unintentionally set to values that maybe hadn't been thought about or aligned. And if we do this a little bit more harmoniously, I think it'll go like the structural and DTI analysis that have gone so well. For example, you could do cross-disorder comparisons more effectively. You could do normative modeling or lifespan modeling, but that does rely a little bit on us all coming together and making decisions, hopefully empirically based decisions. And so that's the reason I think Clara and Leah and all of you have been doing this iterative process of surveys, dialogue, empirical experience to work through what might be a consensus workflow. I do want to say that in deciding a consensus workflow, it's not meant to stifle efforts to try other methods. But I think we would all agree that having some harmony in the pre-processing and goals would benefit us all. So I don't know if with that quick intro, Clara and Leah, you would like to take it away. I think this meeting will run about an hour. But there's so many experts on this call. I mean, certainly this isn't the end of the discussion. So do chat any thoughts or advice, and we'll make sure to listen and work through everything that everyone has to say. So thanks again, Clara and Leah, for taking the lead on this.
Well, thanks a lot for this introduction. So I will start by sharing my screen. Okay, so can everybody see our slides?
Yep, looks good.
Yeah, great. All right, so we will start
Actually, I'm sorry, Clara, to interrupt. It's not in full screen mode. It's in that mode where if that's fine. Okay, that's
better.
Sure, that's better.
Perfect. All right, so we will start by giving you a summary of the result from the survey. So almost like 60 people filled out the survey, so it's pretty good. Then we will provide you the recommendation we did with Leah and Ilya and HALFpipe team for like new resting state fMRI ENIGMA analysis. Then we will explain you the next steps about like any HALFpipe upgrades, any new features, and what we are going to run for to test the new function and everything. And then we will end with an open discussion if you have any concern or any question. All right, so about the survey. So as I said, 60 people filled out the survey from 33 working groups. So it's pretty good because like almost every working group is represented by at least one or several people. Half of you were considered yourself as users or as expert. So it's also pretty good to have like both sides. So half of you were running or planning to run fMRI analysis or coordinating multi-site fMRI analysis. And a third of responders have already processed their data with HALFpipe. And the other third did it with another software. So it sounds like everybody is pretty good with fMRI, pretty up to date with fMRI. So, and that's really nice because most responders would be willing to reprocess their data with HALFpipe. That's really important for us. We have almost half of the responders who would be able to share their individual level data. And so finally now so that was about like who everybody is and like the knowledge about fMRI. But so now we ask you like those questions about pre-processing and processing steps. What do you recommend? What's important? What's not? What has to be done? What can be skipped? So one of the most complicated questions, which processing step have the greatest potential to change the result? So which are the most important to be harmonized? So in red, you have the answers from the experts, and in this blue-green, you have the answers from the users. So it looks like everybody, like expert agrees that confound removal, motion scrubbing, and GSR are critical steps that have to be harmonized. It doesn't mean they are like the most important. It means that they are like the most, like they are critical because they will change the result if we don't harmonize them across groups. User seems to agree, even if like they didn't consider motion scrubbing and GSR as important as experts. So we'll come back to each step and individually. So for temporal filtering, it was pretty easy. Everybody agreed it's important. So at least it provides benefit, or it's a required step. So nobody said it's not, it doesn't have to be done. So it's a step we have to include anyway. For the high pass temporal filtering with 128 second cutoff, everybody almost agrees that's a sensible recommendation, except a few people. And it's good because it's what has been used by the MDG, PTSD, OCD working group already. Slice timing correction, same. It's pretty easy. It was pretty easy because like everybody agree it provides benefit or it's required. So that step is not a problem either. Spatial smoothing, 6 millimeter. So almost everybody agree it's a valid choice, and it's what has been used also from the MDD, PTSD, OCD groups. All right, so now motion scrubbing. So it's currently not implemented in HALFpipe, but the idea is that we want it in HALFpipe because if you look at the result, almost everybody at least agree it provides benefit, and many experts and users agree that's required. So that's going to be included in one of our multiverse pipelines that we propose. GSR, so that's global signal regression. That's a tricky question as always. So people disagree on that, and that's what we already knew from the literature, from talking with people. Nobody agree on that. So it's interesting because experts, like some of them, I mean, half of them said it should never be done, and the other half said it provides benefit or it's required even. So it's really interesting. So it sounds like, I mean, we kind of knew that and already discussed that in our previous meetings, but we have to include GSR in at least one of our multiverse pipeline.
Hey, Claire, I think this was a litmus test for the representation of your sample. So if you didn't get this, I'd be worried. So this is perfect, actually. Yeah.
Yeah, exactly. Thank you. So susceptibility distortion with correction with ENIGMA. So everybody agrees that if you have it, you should do it. So that's pretty easy. And if you don't have it, you don't do it, but it's okay. All right, so that's one really tricky slide. What are the valid confound removal methods? People agree that simple, which is motion parameters, like simple in nylon. So motion parameters, their derivative, the squared motion parameters and their derivative is a valid choice. So it was not done. It has not been done so far by the working groups in ENIGMA, but they used ACOM core, so which is a top 5 ACOM core components. So it has been used by the MDD, OCD, and PTSD. So it sounds like an important pipeline to keep using to be able to compare with what has been already published by these different ENIGMA working groups. Then IC Aroma and ACOM core. It also has been used by these different working groups. It seems valid for most experts. Then only IC aroma, simple, which is that one basically plus GSR, and aroma, so that one plus GSR. So, okay, can you still see my screen? I don't know. So
Looks
yeah,
fine.
okay. Okay, thank you. So it seems
Can
like
I make
simple.
ask one question here? Because we're now talking about the analysis per se, but we're doing resting state as well as task-based fMRI. And for task-based, I would agree simple would be just okay. But especially if you're looking at connectivity, I would not agree. So how's that taken into account here?
So here we really build those guidelines for resting state 3, not for task-based. We will do a different guidelines for task-based specifically.
Okay, thanks.
All right, so then Leah is going to dive into that a bit later, but we just wanted to mention that IC aroma and ACOM core are conceptually very similar. It's basically like dimensionality reduction. One is based on ICA and the other one is PCA. And we talked with several experts and they told us that's quite redundant to do both at the same time. So basically aroma plus ACOM core. So we decided to not include it in our multiverse pipeline, but Leah is going to show you a bit more about that based on Violeta work from HALFpipe team. So QC. So it was pretty easy because almost everybody agrees that we have to do visual QC, and we have to exclude subject based on FD, framework displacement. The dice coefficient index for the overlap between the pre-processed full like brain mask and the MNI template. Most people didn't know about that, so we're probably not going to implement that for now, but maybe in the future. But at least visual QC and FD threshold is something we can agree on. We did agree on. So the LTS, the version of fMRI prep. So for now, as you know, fMRI HALFpipe is running the LTS version. So the one released in 2020. So we talked with the fMRI prep people. So they are going to release the new LTS soon. So we might have to update it. I mean, we will update HALFpipe with the new fMRI prep version. And for now, we'll keep that one. It will require a lot of work from the HALFpipe team because they will have to verify that the results are not affected, or if they are, like, we have to know how much they are affected. So that will take some time, but the new LTS is not released anyway. So using consistent version, everybody, like 2/3 of you agree it's important. So the last part is about like which processing step we want to, I mean, which feature we want to extract, basically. So everybody agree atlas-based connectivity is important, it's a valid choice. Expert and user agree on that. And then it was not really clear which other metric is important to extract. So it's, I mean, Leah can go back on that, but we decided for now to not include RIO and FLFF because it requires different pre-processing steps. So we can discuss about that, but we wanted to reduce the number of pipeline we have to run. Then after, obviously, each ENIGMA working group can run whatever they want, obviously, what's answer their question. But like for the common base, we wanted to don't ask people to run too many things. So only atlas-based connectivity, seed-based connectivity for subcortical material, insula, and cingulate area, because we asked you in the survey which region you would be interested about for seed-based connectivity, and that's the one who came back the most. And network-based connectivity. So also Leah will come back on that.
So that will still be available, just not required. And
exactly, you can still run whatever you want.
And when you say it requires different pre-processing, you mean the different temporal filter?
Yeah, basically we decided based on the survey. So we'll show you, we decided about pipelines, pre-processing pipelines, for example, with or without GSR, or like, but if you want to run Rio or FLFF, you cannot do the same scrubbing, the same temporal filtering, so you have to do specific pre-processing pipelines. So we would have to add another version. So at the end, we will have like 5 or 6 different pre-processing pipeline, and we decided it's kind of too much. So we wanted to reduce the number of way of processing the data.
Would it be possible to have a canonical FALF-RIHO pipeline, even if it's not required of the groups? So and the reason I ask is I think this is going to be one of those ones where people are going to want these indices anyway. And for the people who do want it, I would love to have it be harmonious across groups.
I think the issue there is, Claire, correct me, is that we, I don't think we have a strong sense for what that canonical processing pipeline would look like, given the expertise, the feedback we've had, right, Greg? So I mean, certainly we could create something, but it would be perhaps what you would want, or, I don't know that we have the input, I guess is my point.
Right. I mean, we could, I think between us, we could solicit that input if we wanted specifically to have that. But my impression from what I've seen from people is if we just don't do that level of temporal filtering, that's the big one that's going to interfere with FALF and Rio. This could be a later discussion.
It looks like Leah and Jessica want to say something.
I just wanted to second Greg's idea of getting an ALF pipeline. I think that would be a tremendous boon, but again, not necessarily the top priority, but I wouldn't want it to drop off the list either.
Okay.
Yeah, I was just going to say, I think we should add it. I think it's a good idea. We just want to keep the main recommendations as lean as possible so as to not discourage groups from running too many things. Because if the project doesn't relate at all to frequencies and things like that, then of course it might add more complexity to a project. So we wanted to keep these recommendations as small as possible, but I guess it might be nice. I agree to have some additional things in there.
Okay, so I think we can continue. So yeah, I just have one slide about these atlases. So we ask you which atlases are considered as valid. So it's interesting because basically it sounds like people really know like Schaeffer 400, DAO, and free surfer subcortical, and all these new atlases like the DFIMO, the cerebellum atlases. So almost all of you didn't answer these questions about those atlases. So it sounds like we will continue with the Schaeffer 400, DAO 17, Buckner for the cerebellum, and free surfer for the subcortical. So in HALFpipe, Leah already did one Schaeffer 400 like combination. So it's like already all together. So it's considered as only one atlas. And in the future, for gradient analysis, we also want to add a surface-based atlas. And about that, now we have a few slides from Sophie. So it looks like you want to.
Yeah, I also wanted to say something for like the previous slide. Like I also emailed, like I would very much encourage to have automatic output of more than one atlas, just to know that certain results are kind of robust and not a Schaeffer 400, especially Schaeffer is asymmetric. So certain comparisons cannot be made between left and right. Buchner is one way of looking at the cerebellum, but there's also like a lot of movement in that field on like cerebellar subregions and what is more relevant. So I would at least have one structure on one functional atlas at a similar granularity level. Or I don't know, like I know that reviewers always ask, like, can you also show that it doesn't matter what atlas you've picked? So I would think it's good scientific practice to have at least this atlas replication built in, but like, I can also understand if this is my personal preference, right? And it may also complex things, but Frank is also raising his hand.
Yeah, Frank, if you want to.
Yeah, I think that's absolutely where we're going. Sophie, that's clearly a goal to have more than one atlas output. And there's others here not represented, Glasser and Gordon and Power, and I mean, it goes on, right? So, and subcortical versus cortical atlases, there's some differences there. So all that is challenging, but definitely having more than one atlas, I think, has to be a standard.
I just wanted to add real quick that one of the problems we run into is that many of the atlases that are recommended commonly, like Glasser or Gordon, are on the cortical surface. So this is something that HALFpipe currently does not support. It might be something we add a bit further down the line and start with these simple recommendations, although, of course, yeah, it might be nice also to add in the atlas right away. So this is a great discussion topic. Sorry, Raj, I didn't see your hand was up.
My hand went up after you started talking. Thanks, Leah, though. I mean, I think I agree with Sophie and everybody else who's mentioned about other atlases. I guess the question in my mind is, even if we say Schaefer 400 is the sort of agreed upon harmonized thing, if you want to do a second atlas, could that second atlas for confirmation or whatever be the choice of the working group or the project? And does the second confirmation atlas have to also be harmonized like the Schaefer, which is the primary one? I guess that's my dilemma. It seems like, well, my hunch is that the second atlas doesn't have to be harmonized, but I don't know. I just wanted to throw that out there.
If I could jump in real quick, yeah, it's a good point, Raj. I would see the harmonization as happening pre-atlas to help make it, so you have this sort of homogenization of signal or whatever you call it at the level of, so that you combat for your correlation matrix. Or yeah, no, you're right. Actually, at the level of the correlation matrix, atlas would matter. No, I think you would have to harmonize for all atlases. You wouldn't want, otherwise your comparison ends up being sort of I mean, comparing between atlases is somewhat apples to oranges anyway. And in some cases, it's like, apples to pianos. But I think if you harmonize the one, you're probably going to have to harmonize the other, unless there's a step you could do pre-correlation matrix, and then all your data are harmonized. Now you move it into an atlas, and I'm trying to think of what that might look like. But yeah, it's a really good point.
Any other feedbacks about that? No? All right, so now we have like a few slides about introducing gradients from Sophie. So I think Sophie already presented in several working groups calls, so you might know about it, but I think it's good to have a quick reminder about gradients. So if you want to, I can move your slide if you want.
Okay, so yeah, I'm Sophie, and we were thinking that it's also relevant to take more of a system level perspective, so beyond ICA and other measures, in terms of standardized output for ENIGMA. So as some of you hopefully know, we can differentiate in the brain between sensory and association areas. Maybe click on this, Clara. And this differentiation can differentiate more perceptual action functions towards attention networks, towards more social cognition functions. And what is kind of relevant here is that it puts it in kind of a spatial differentiation. So you can also see the distance between networks. So it has a certain topology and organization. And we think this is highly relevant because it can also be linked to anatomical hierarchy, then ultimately also helping for a mechanistic understanding of how function could relate to structure, and also in clinical relation, like what could be underlying it, but also in terms of evolution. So getting more to a systematic computational view on how the brain is organized, and also where and how we see these changes occur along a gradient in specific developmental milestones or disorders. So next step, please. So luckily, there is an out-of-the-box, already usable toolbox with BrainSpace, where I have also a QR code in a bit. And also we were thinking of implementing, or we wish to implement this in HALFpipe. So the simple thing is you put in an input matrix. This can be based on either volumetric-based time series that are correlated, but surface-based can come with an additional benefit of alignment if the data quality is good enough. But I'm also a pragmatist, so one can think about what works best for you and what you want here also. So once you have this input matrix, then you do an affinity kernel function to normalize in the next step also. And then the next step, you do a nonlinear decomposition of the signals, creating either with a simple PCA, like different dimensions, but you can also do a nonlinear decomposition based mostly on the top 10% connections, but also here depending, like this is a standard framework, which you will use, and then you get like this nice differentiation between sensory and association regions, but also within sensory regions. So here you see also sensory motor and visual areas highlighted, and then next enter, and then you like resulting, you will see that regions and their connectivity profiles are sorted along this axis, so to say. Next slide or enter. So voila, so if you want, you can make a photo or whatever of this QR code. But you can also photo like for later. So next slide. So why is this relevant? Like, for example, take autism spectrum conditions, and here we used ABIDE, but similar things could be done in like all other disorders. So people have also done this for schizophrenia, where we have looked at how this differentiation between sensory and transmodal areas is maybe different in people with ASD relative to non-autistic individuals. And indeed, already looking at the patterns here, you see like this marked changes, like I cannot annotate, but you see the default mode network here in red, which is much more pronounced in controls or non-autistic individuals than in autistic individuals, meaning that these regions are more differentiated from sensory areas in non-autistic individuals relative to autistic individuals. And if you do enter, you also see the statistics on this. So what you actually do in a way for people using cortical thickness data is you can create a kind of cortical thickness level of way thinking about the brain using functional data. So it's a different way to make maps. And for me, it's a very intuitive way to understand also how local regions and their connectivity change relative to each other. And this can also have clinical relevance and also like help with more mechanistic understanding of how these organizations and axes may be shaped, which each axis also mean a different dimension. So here it could link to neurodevelopment. So this paper describes it way better than I pitched it here, but we think that by including this in the pipeline, it would open up a lot of like more theory-driven accounts of like topological organization of resting state networks, which may have a relevance to various disorders that we study here in ENIGMA, including especially neurodevelopmental disorders, but also neurodegenerative disorders. So this may be very relevant and also is done in the general focus point of the ENIGMA gradient working group. Yeah, finito. But for questions.
Thank you so much. Thanks so much, Sophie. Any question about that? No, then we have like, so everybody agree to stay up to date about those things. All right, so now we will, Lea is going to present the main recommendation for future resting state of MRI analysis. So Lea, do you want to share your screen or do I continue to?
It would be great if you continue. I don't have it open right now. I had some browser problems earlier. Yeah, so it's really cool that we were kind of able to all come together with the survey, which I think is really wonderful. So this is kind of just a summary of the decisions that Clara just presented. So we kind of have a set of common pre-processing steps, and as many of you might have noticed, we kind of also phrased the survey in a bit of a leading way. Because we, of course, wanted to be open to all the results, but we also wanted to just confirm that what groups have already done is kind of valid. And we got good feedback for slice timing correction, that this is kind of a valid step. We got good feedback that the field map correction that many groups have already been doing, and spatial smoothing, and also that we use the Gaussian weighted temporal filter. So this is the standard one implemented in FSL. So this does not take frequencies, but it takes a Gaussian smoothing approach to the time series, which is very nice. So that means we kind of have those as a general basis that we can build on. And we have not gotten feedback that, yeah, anybody in the community and across the experts that we surveyed think that this will greatly affect results. So this is already very nice to see that we have kind of a common basis to build upon. But now, of course, there's the steps that people don't agree on. And this is great because this really gives an opportunity for all of the papers that come out of this to also answer these methods questions that have never really been answered and that lead to low reproducibility in the field. So it's kind of like, yeah, feeding two birds with one stone in a way. The first pipeline is the one that was used by the MDD and PTSD groups, and I think it was also used in the OCD group. And it contains just a COM-core denoising as the denoising method. So, but this has been proven it's a good method. The other two methods are a bit more advanced, so they will both include motion scrubbing, which I have grayed out here because HALFpipe currently does not support it. So our goal with HALFpipe was kind of, yeah, to have like a nice user interface to fMRI prep to make it easy for people to calculate all these things and to get everything ready. But the focus from the start was a bit more on task-based, and resting state is something where we never really, until now, put in this much effort. So I think adding motion scrubbing is going to be a big step towards that. And so we want to support that by September so that people can get started with these projects with all three pipelines. But pipeline 1 is already available right now. And so that means kind of any projects that are kind of like aiming to start very soon can start with pipeline 1. And anybody who's like planning a project can do all three pipelines later. And so then these pipelines 2 and 3 are identical. They both include motion scrubbing and regression of the 24 motion parameters that Clara listed earlier. And then 3 additionally contains global signal regression. Yeah, so once we've calculated those preprocessed images, we will calculate different features. So the most important one, as Clara highlighted, is atlas-based connectivity. So here we have the Schaefer combined is what I call it. We can also rename it, but the idea is it's the 400 reset region 70 network Schaefer atlas combined with the Buckner cerebellar atlas and free surface subcortical atlas. So that means we get whole brain coverage in just one connectivity matrix. But if you just want the Schaefer regions, you can always, for example, for cortical gradients, you can always filter the matrix and take out those columns and rows you don't want. Then we also want to add, once we have surface-based processing implemented in HALFpipe, which is high on our list, want to add the Glasser atlas in and also combine that with subcortical and cerebellar atlases. And this will be like the opportunity to use more modern atlases and add more things, in my opinion. But of course, this is a discussion and we want to basically accommodate everyone's needs. So adding more stuff already now is also a great option as discussed earlier. And so then with the goal of implementing gradients, which I also put in gray because we haven't done that yet. Then the other feature that we kind of thought was easy to include was seed-based connectivity. Yes, Greg, you have a question.
Yeah, just I heard why you didn't want to do a comp core along with ICA, but leaving a comp core out of pipelines 2 and 3 means that we don't have any pipeline with both motion regression and a comp core, right?
So pipelines 2 and 3 are a bit more focused on the motion aspect, but so the idea is kind of it's 2 different approaches, right? So a comp core looks at regions of the brain that are supposed to only contain noise. There's noise. There's no like somata of neurons there and so on. So we wouldn't expect anything there, whereas these ones use the motion parameters. So it's kind of different approaches, but recent empirical papers have shown that they have similar properties in terms of what variants they remove and how the matrices look in the end. So it is quite likely that the overlap will be significant, even though they use different approaches. Although it is very much an empirical question, and it's always going to be a bit arbitrary. So this is very much based on trying to basically take the winners from the survey and also talking to experts and looking at recent papers. So it's very hard to make a choice here. And of course, it would be nice to have more variations with a comp core. But I feel like this would be a focus for more methods-focused projects that I'm certain will happen. Because that's also one of the focuses, I think, Frank, of what you're aiming to do, right? So that would be an interesting thing going forward.
Absolutely, yeah.
Okay. So for seed-based connectivity, to keep this short, we wanted to focus it down on a few regions only, so subcortical regions, insular and cingulate, so that we can have kind of the main networks. And finally, one thing that we've been wanting to add is also network templates, so looking at brain networks with NeuroiMag, which is a constrained ICA approach that we're aiming to implement also soon. So yeah, this will be also available in the recommendations. So, and then as we decided, all of these will be combined with visual quality control. So kind of the idea is that HALFpipe will run all of these things automatically, and then quality control happens afterwards. So you can decide which ones of them to share with your central site and or include in summary statistics calculation. And so we will apply a mean frame with displacement cutoff, and we will apply visual quality control in the recommendations. Yeah, so that's kind of the things. It's a very complicated slide suddenly, now that we have everything. But at least we have like a process that is kind of shared.
Yeah.
So
and just to add, like we will provide you like written guidelines, and this like figure is going to be part of it anyway. And we will summarize all this survey. So we are working on that, and we will provide you this document soon.
very cool.
Yeah, so if you want to present the new team.
Of course, who will work on this? So it is the new team of HALFpipe that is working with Ilya Weer in Amsterdam, and they're all here in the call. So I just asked them to introduce themselves. Tomasz, you're first.
Oh, well, hello. So yeah, that's me. I'm the first one, and I'm mainly now doing the things that are listed below my photo there. Yeah, and I guess that's all.
Maybe say a bit about your background, what you did before, what
Yes.
how long you're on the team, what you're going to do.
Yeah, so well, basically, I did PhD in astroparticle physics and experimental. So there was a lot of programming, coding, and data analysis. I'm still waiting for my defense though, that is in September. And yeah, I'm in the HALFpipe team since December now.
Well, welcome,
I-
Tomasz.
Oh, yes, thank you very much.
Hey, Violetta.
Hi. Yeah, I also joined in December. I am I graduated from a research master in brain and cognitive sciences, and I also spent a lot of time programming. So I thought this project was perfect, and I am working on implementing consistency checks. So when we upgrade the fMRI prep version that I will also lead, we can see exactly what's happening with the results. And actually, Lea is going to present some of the visualizations I've built to compare two versions of the same results, basically. So yeah, that's me.
Yeah, so Violetta prepared a lot of slides, so we don't have time to show you a result, but she's testing the consistency of the different ICR and the different method, and also like she will test the different between the different fMRI prep versions. Sorry.
Fantastic.
Yeah, stay tuned for the next meeting. Finally, Nadja.
Hey everyone, I'm Nadja. I think most of you know me better from the ENIGMA OCD group. I'm a PhD student of Odile and Ilya, and I'm working on task-based data, so kind of the extension of these resting state analyses, but then really with task-based data in different cognitive domains in people with OCD. So I've been using HALFpipe for a long time, and I've been supporting some of you, but I've more recently joined the HALFpipe team officially, as sort of more for user support, so to help develop the documentation, to help make the user experience even more smooth.
Oh, that's fantastic. That's very much needed. So.
Hey, very cool. Thank you guys. So now I'm going to present some of the work that I really didn't do much on, but the idea is kind of to talk these last 3 or 4 minutes of the talk about what our next steps are going to be. So of course, as outlined before, we need to add motion scrubbing. And that's not an easy task because we need to make it compatible with all the other steps that we do. For example, if we scrub out a volume in motion scrubbing, we need to make sure that temporal filtering is not just going to average that right into the adjacent volumes. We also need to make sure that confound regression knows about those volumes that we took out. So this is not a big deal to implement, but it has to be done. Same for upgrading the FMRI version and other things. Yes, Frank.
So I hate to, I know this has been resolved, but motion scrubbing creates so many problems for data. And there's a number of papers that show that it reduces the reliability of your findings. We have really good data for that coming out now in a paper we're sending out. Unless the community really wants it, I mean, I think it's something 5 years from now, very few people will be doing. But if we need it, it's going to be work to integrate something that I think creates problems for your data. So I'll leave it at that. I'm only one voice. I know we have a lot of experts here, but I'd hate for us to go down a path where because this will take some work, right?
Yeah, exactly. I mean, we have to focus on this. I guess maybe we'll keep the discussion on motion scrubbing. I think it's a really valuable discussion. Let's keep it for the end so that we can just get through the slides. And then we have lots of time. Sorry about to cut this short. But yeah, so I guess the thing is, if we're going to make changes to the workflows in any way, next slide, please. What is kind of important is that any changes we make may lead to different results, whether it's changing the FMRI version or adding motion scrubbing or not adding motion scrubbing. It could, do things. So Violetta has been working on implementing a consistency check so that we run a subject, first of all, repeatedly to see that is there any stochastic random variation. Then also looking at different versions of the code, are current versions compatible with past versions and so on. And also after making changes to the code, like adding temporal filtering or motion scrubbing and so on, making changes to that. So this, I think, is a really important thing that we really want to have before we make these bigger changes, because only then we can kind of guarantee the most important aspect of this, which is reproducibility. What have we found so far? Next slide, please. It is that Violetta has run one subject. So this is, for now, it's only one subject twice. And so what we see is that when we run it with the ACOM-core pipeline, which is the one that we also want to put in the recommendations, there's basically no difference between the two runs. But with ICA aroma, because ICA is inherently based on, well, a fitting process that is randomly initialized and has very many decision points in the algorithm, we see big differences even in the final connectivity matrix that we get out. If we repeat this again and again, and this is something that we can also show in a more detailed setting if that's of interest. These differences, if you try this again, will be in a different part of the brain. So it's kind of very, very stochastic. And so that's also why we are not super happy about IC aroma and really think ACOM-core might be the more reliable and robust option going forward. So this is really interesting. And so now the goal is kind of to run this for more different subjects so that we have a kind of more general representative idea of where this variation comes from. And then also to implement this in a way that kind of automatically runs when we change things in HALFpipe. Next slide, please. Tobash has been working on a new user interface, as he said. So as you know, the HALFpipe user interface is very bare bones. It's a very linear thing that asks you questions about your data. But this will be still in the terminal, but like a completely modern user interface where you click on things, you can select things with your mouse or with the keyboard. You have visual toggles that indicate, and it's most importantly not linear anymore. So you can jump back and change things after the fact. And this will also make the code more easy to use and easy to extend with things like motion scrubbing or other things in the future. Next slide, please. So from the survey, we got a lot of suggestions for other stuff that we might add, and all of these are a lot of work. So most importantly, using existing FMRI prep outputs, I think would be wonderful, also to avoid kind of the computational cost and environmental impact of having to rerun those things. Of course, compatibility with different templates and different data types, supporting more outputs and analyses are really important. And of course, also, making it more nicer to use in yellow on top are, I think, also very interesting. So if any of you have time to support us also implementing these, HALFpipe is an open source project, and I'm really happy to chat and get you guys on board, helping with the code and working on this. And we're also going to try to put these somehow in our schedule. That is very full, unfortunately, but we're trying to do these as soon as possible. Or if possible. So yeah, so that's, I think, my part. So I guess we're going to go to the open discussion. Thank you so much.
Thanks so much, Leah. So yeah, I think we have 10 minutes for the discussion. So yeah, Jessica.
Just a quick question, because the Enigma schizophrenia group is just getting started, and we literally just rolled out our manual on how to use HALFpipe and what we were going to do. I'm assuming that as you update this over the coming 6 months to a year, the older version will remain and not be broken, hopefully. Because we're going to want people to, it's going to take us a year to get everybody to really run everything, and we need to be stable, and we can't put it off, for another 6 months or something.
Yeah, so everything that is right now in HALFpipe will remain. And if we change something, we will have a consistency check proving that results are compatible. This will be for at least 4 different subjects from 4 different datasets from diverse scanners. So that means we are also planning to have this in a paper and to get it peer reviewed, so that we kind of really prove these things as much as is realistically possible. The new user interface, and Tomasz, if you want to go into this, it's going to just be a new, so we kind of have a UI part in the code and we have a new UI part in the code. So it's kind of like they will just both be available. So it's, and yeah, at some point might remove the old one, but there's no plans on that for now. And of course, any versions that are right now downloadable will stay downloadable for, yeah, basically forever until we run out of money to host them.
All right, thank you.
Though it would be good, maybe to add, it would be good to use the settings we now got out of the survey to already incorporate them in the manual for the schizophrenia working group. So the atlases, the seats, etc., for cross-disorder comparisons.
Any other questions, things to discuss?
Yeah, just one thought is that as Tomas's group is working, I think for me, the biggest thing that somebody could improve about HALFpipe is the error messages. Right now, if we get error messages, I have to bother Leah, and finding a way to not do that would be lovely.
I guess I have a really quick question, and thanks, Clara and Leah, and everyone who's contributed to this. It's actually phenomenal that we're at this point. My question for the group, and this might be for the clinical chairs and people leading analyses at the moment, is how these recommendations should be used. And so, I mean, a recommendation could be a gentle recommendation that people proposing a project align to some degree with the recommendations. Or, I mean, certainly disorders that plan to compare their results directly, and there are many. There are even grants comparing PTSD to MDD that Raj and Delin and Jenny are leading. So I'm interested in this sort of meta question, which is, should these be, I mean, they can't really be enforced because much of the work is voluntary, but to what extent do folks feel that this is, a valuable exercise? It should be run by people proposing fMRI analyses, or perhaps even a stronger recommendation. I mean, please do these if you plan to do anything else. I mean, please do these as part of your analysis, and then by definition, your results will be comparable to all the other work. I mean, I'm curious about this sort of overall question of how these recommendations should be rolled out, used, implemented, modified.
Yeah, I may have at least just my own sort of perspective on that, Paul. And I think part of it is every lab needs to be able to do what they're going to do. But then I think the carrot for the lab, all labs, to participate in this sort of community effort in particular is if we can make HALFpipe user-friendly enough and computationally inexpensive enough so that labs end up doing both. So maybe they run their own analyses, but there's a lot of incentive for them to just run their data through this pipeline so that they're available, that they can contribute to the community when called upon or when a collaboration is possible. And I think that's going to be a ton of incentive to be part of these projects. Now, maybe I'm wrong, and for some labs it will, and some labs it won't. But I think that people on this call, they're on this call because they're part of this community and they believe in this sort of the team science. I think people outside this call may be a different, that may be a little more challenging outside of Enigma. But in the end, I mean, I think it comes down to, can we make it easy enough for labs who perhaps don't have all the resources that many of the labs here do, you know, as far as personnel and expertise, can they run HalfPipe in a way that they really are ideally want for their grant, for their aims, but then also have in their sort of back pocket, be able to run the recommended canonical pipelines so that those data are ready, or those data can be used to contribute. I think that's really ends up being the question.
Guido, if you want to add anything.
Yeah, I largely agree with everything being said. I'm a bit puzzled with moving away from the typical pipeline, pre-processing pipeline, I have to say, what has been used. If the first working groups have all used something but scrubbing, I think it would be a pity to just then change it all together to scrubbing, especially if others kind of question whether you should do it at all. My experience is that people don't care much if they just want to run it once and don't want to be bothered twice. They don't care for how long it would take, so they would probably be happy in running multiple things, especially with respect to output. So I better do 10 different atlases than choose just one, so people can do this later on. But the same doesn't hold for pre-processing, because if you would want to run different pre-processing pipelines, it just explodes basically storage. So I think you have to make a decision on the pre-processing pipeline, then provide as much output as you can, so that it will hopefully be backward and forward compatible. And then with respect to pre-processing, I would prefer to keep it comparable to what has been done in the initial working groups, but that's of course up to you.
Yeah, so I totally agree, and that's something we discussed a lot with the HalfPipe team. So it's why we wanted to keep also the ACOM core pipeline, which is basically what has been used already. And then we decided to add those two additional pipeline, because it sounds like in the survey scrubbing seems to be important for most people. So it's why we decided to add one version with scrubbing and GSR. I mean, if we don't do GSR, it's going to be asked by reviewers anyway. So I think it's still important to do at least one version with GSR and one version without. So that's why we wanted to reduce the number of possibilities, the number of pipelines, obviously, because as you said, like then you have like it requires a huge amount of storage, so we cannot afford that. But we ended up deciding those 3 pipelines were the minimum to keep one version from ENIGMA, what has been done, and what else. But I mean, I don't know, Leah, if you want to add anything.
I mean, I was going to add, I mean, I'm totally on your page as well. I think the issue we're running in, that's kind of a compromise, right? That for many people, having like a long list of recommendations can be a bit daunting. So kind of that's why we kind of went out with a survey, and we saw that for many of these atlases, the majority of people said, oh, I've never heard of this. I don't really mind that atlas. So it's kind of like, if we first do the survey, then we kind of also have to be happy with the results in a way. And that's too bad, because I also don't personally I'm not a fan of motion scrubbing, but we're going to, yeah, it's kind of the difficult kind of situation we're in now, because of course finding a compromise and finding a consensus is never going to make everyone happy, and that's the difficulty. So the basic goal of this project was to find kind of like a minimal set of things that we can ask everyone to do without them being like mad at us for forcing them to do it or asking them to do it, which I think we've accomplished. Now the question is, what do we have to add to make this useful? And I think what we've gotten really as a feedback in this is that we want to add more atlases, and I think that's a good idea. The question is, do we want to expand what we do with ACOM core? Like, I mean, these are discussions, and I mean, of course, we can have, but it's also important, I think, to just say, well, we did the survey and now we got the answer from the community. So it's a difficult situation, of course.
Yes, Frank.
Agreed. And Leah, sorry for throwing the grenade into the middle of your presentation on scrubbing. So to Guido's point, which is really important, maybe we could just take a 10-minute resting scan and run it through the 3 recommended and just see how much space we're taking up with that. If time is not the typical issue, let's just get an idea of, is it way too cumbersome, or is 3 at what point do we reach threshold where groups are going to say, forget it, I can't process my data in that many ways, and I don't know if 3 is, maybe 10's too many, maybe, but so that I'd be happy to take that on. We have some data we could, with students here, we could run that and just see, and then we could have some numbers posted. Here's what you're up against data wise, storage wise, time wise, if that's helpful.
I think that would be wonderful. Also to get some initial feedback. I mean, I know you've been running lots of multiverse things, so I guess it would also be really cool to see how they compare. I mean, even if it's just what we've seen in the slides, like a difference between the connectivity matrices, or something like that, it would be very interesting to see. But yeah, I mean, I guess the idea is that since we're not saving the entire preprocessed image, it's going to be not a huge amount of space. So adding another atlas is going to be 10, 20 megabytes per subject, which I don't think anybody will revolt against. Adding another pipeline will be about 100 megabytes, but it would be great to have numbers for this.
So I think we are almost done. So we'll probably organize a new call in September. We can present you any updates. And before that, we'll circulate the first version of the guidelines. And in September, we can update you on what's been implemented and what's the next steps again. So we want to have regular call anyway, so every 2 months or something like that, to be able to address any issues. So yeah, I don't know if Paul or Leah, you want to say any final words.
Are you just hugely appreciative to you, Clara and Leah, and all who contributed? I mean, I think what you said as the next step is a good one. You've made this an act of consensus and diplomacy, and so I think you've included all the viewpoints that weighted by the level of expertise that people have. But I think, as you said yourself, Clara, I mean, if folks have other recommendations or comments or feedback, we can't get through all of this in an hour. So, I mean, do reach out and put it on the agenda for the next call. But I just want to thank you for bringing some logic to the anarchy. It's
Agreed.
been
Yes,
very
thank
helpful.
you.
Thank you. Bye. Have a great day.
Thank you, guys.
Have a good summer, everybody.
Goodbye.